this shouldn't hurt your biology career. But if you have any questions over the chapter that we didn't go over and you just are curious, feel free to come to me. Okay, so this is on cell communication. We're going to look at how cell, yes?
Before you start, I wondered, do you input this in the exam altogether, or just when you get through today?
No, whatever we go over today is material for the exam. What I'm saying is there's a lot more material in the chapter in the book that I'm not going to require you to know. Yeah. And we are having a review Wednesday, so if some of this isn't clear, please bring questions. I have a review set up for you guys, but I want y'all to bring questions, of course, to help me know what y'all aren't getting. Okay. Okay, so we're going to look at how signals transmitted, ligand binding, signal transduction, and cellular responses, components of signal transduction pathways, how a cell can respond to a signal and fine-tune a signal, and the importance and mechanism of apoptosis. So apoptosis is cell death, right? Okay, so the communication in our body between cells is really as simple as how we communicate to each other, right? If I need my boyfriend to bring home groceries from the store, I first have to tell him that, right? I have to send a message. So if I want to get on my phone, right, and call him, I have to first send that message. The second part of communication is that he has to receive that, right? So even if I leave him a voicemail and he never gets it, he never receives that message, is he going to bring me groceries home? The ones that I wanted? No. So that's the second part, right? Receive the message. Okay, the third is going to be a response, right? So our body does the same thing. We have cells that send out signals, other cells that have very specific receptors to receive those signals, and then they carry out different responses. So if you think about it, right, the receiving is very specific. I'm going to dial a phone number. It's not going to call anybody random, right? That phone number goes directly to the person I'm calling. And these signals that our cells send out have very specific receptors that receive them too. Okay, we're going to look next at how these cells communicate by sending and receiving exactly. So a lot of times we look at microorganisms and how they communicate, which reflects how our cells communicate on a broad concept. So these are yeast cells that are different strands. And how these work is that they have these receptors on their cell that are specific to other types of signals. So they don't want to mate with the same type, just like inbreeding is bad for humans, inbreeding is bad for most organisms, okay? So this blue cell here wants to mate with a different type. So the signals on its, the receptors here are specific to other types of signals. So it releases factors that are round that are not going to bind in its own receptor, right? This one over here, this pink cell, is going to send out signals that will bind in the receptors. Let it know, hey, I have different DNA than you, we're a good match, okay? So once these signals bind into these receptors, there's a relay of messages that go on that will cause a response of growth towards that cell. So fungi, right, or yeast, they live in the ground and they don't move around like animals, but they can grow. So when they receive this signal, it tells that side of the cell to start growing in that direction because there is another type that has different DNA out there. And then it grows together and it's able to mate, if you will, or exchange DNA, and then you get a new wild type or a new genetic makeup of a yeast cell, right? Okay, so just like this occurs, our body is going to have specific cell receptors on our cells that correlate to specific ligands. And remember, a ligand is any signal that binds to a receptor. Okay, so when we look at sending a signal, it can be through direct contact. So this could be cells that actually share a cell membrane. They have junctions, right? And these signals just pass from one cytoplasm to the next. This is a plant cell, right? Go through plasmodesmata. Or it could be, remember, our cells have glycoproteins, oligosaccharides, things like that, that are on the the surface of our membranes and act as recognition sites. So maybe this cell binds to a receptor here through direct contact from one cell to the next to create a response. Okay, but more often than not, it's through indirect signaling, right? So we have our nervous system and our endocrine system that can send out signals throughout the whole body. And it's not a direct, the cell that's sending the signal doesn't necessarily come into contact with the target cell that's to carry out the response. Okay, so the endocrine system releases hormones, right, that can flow through our bloodstream and be received by different cells. So it pretty much, anything that enters our bloodstream will reach all the cells in our body, but only those cells that have specific receptors will actually bind and carry out a response for that signal. Nervous system, right, we have neurons cells that release, they basically transmit a signal through electrical impulse that travels down, and then it causes the release of these neurotransmitters that are received by close by cells. Okay, so you don't have to concentrate too much on this, but there's local signaling where the endocrine system releases these signals in the just the extracellular fluid area, and it's pretty much just a very local signaling. So like when you get a scratch, there might be like a local signaling right here, as opposed, and this is called paracrine signals, as opposed to long distance, right? Hormones that are put in our bloodstream, like I said, go to all cells. And then when we look at the nervous system, a close-up look here, these neurotransmitters are just diffusing across this very small space here called a synapse. I don't really want you to concentrate too much on that. I just want you to understand that signals can be given either through direct contact of cells or long distance through the release of hormones or by neurons, right? Okay, so I just said all this, but the reason this works, right, is because we have specificity in the signal received. Okay, so if a cell does not have the correct receptor, even if that hormone comes into contact with the surface, it's not going to create a response, right? Just like, I don't know, if I call one person's phone in here, no one else is going to actually answer, right? Because y'all don't have a phone to pick up the other end. Or if I send out a text message, right, to one person, all y'all are in here, it kind of is transmitted, but only one person has that receiver, the right receptor. Okay, so once the signal has been received, well, there's 3 stages of the cell signaling and reception, right, to carrying out the response. The first one is reception, where the molecule binds to a cell protein. The second is transduction. So transduction is usually made up of these series of relay molecules that are typically proteins. And what happens, is that when this signal binds this receptor, these are proteins. If something binds a protein, what usually happens to that protein? Changes shape. It changes shape. Okay? So these are transmembrane proteins. So they reach from the outside to the inside. If this causes this receptor to change shape, it's going to cause something to happen on the interior of the cell that transmits this signal to a relay protein here. It causes this protein to change shape to send that signal, to transduce the signal to the next relay molecule, and so on. So each one causes a change in the different protein typically, and it passes the signal on until it gets to the final protein that's going to carry out the response. So this last one here will activate the cellular response. And that can be a number of different things, right? Our cells do all sorts of things. But for example, it could be maybe rearranging the cytoskeleton of the cell to move it, right? Or to turn off or on certain genes. If we need more production of certain proteins, that can be a response. to a specific signal. Okay, so in this whole process, you can't gather, this is something I really want you to get right, is that it is highly specific. So here we have a signaling cell that releases an extracellular signal molecule. Okay, if there are these three cells it comes into interaction with, it's only gonna bind to the specific receptor that it fits with, right? So this is called a ligand, again, signal, and it will bind to the proper receptor that it fits. Okay, so this really helps ensure that certain activities, right, are carried out when and where they're supposed to be carried out. It helps coordinate all sorts of different reactions in the body. So you might have a cell send out several signals that go to several different cells to create a response, right? Okay, you are not responsible for knowing these types of membrane receptors. They are covered in your book, and there are three main types of membrane receptors, okay? And they come in different forms, and they have various different ways of reacting. But all of them are very specific, right? They have a binding site that's exterior to the cell, and then they have the portion of the protein that extends into the cell. So they're transmembrane, and they create some sort of change. One of these we have looked at before. Do you remember ion channel, gated channels, right? That let ions flow through. So this is an example where a signal is sent out, it binds this ion channel and opens it up, right? Allowing those ions to flow through, or it might close. But again, you're not responsible for, they're pretty complicated processes, so we're gonna skip that part. Okay, so why do they have to be transmembrane? Otherwise it would.
Otherwise it would be good.
It wouldn't be any good. Right, basically it has to accept the signal outside of the cell, right, and transduce that signal to the interior of the cell where the response can be carried out. Okay, and we don't really have to go to that. Okay, so there are also intracellular receptors. Okay, so we just looked at ones that are in the plasma membrane. There are some on the interior of the cell, either in the cytoplasm or inside the nucleus. Okay, so if they're inside the cell, what does it tell you about the signal that's being sent? Does it have to enter the cell? Yes, it does. If it's going to bind to the receptor, it has to enter the cell. So most of these ligands, these signals that are sent to cells that have intracellular receptors, are going to be non-polar hydrophobic molecules, typically like testosterone or estrogen or things, hormones like that, that are hydrophobic or really small. That way they can just pass through the cell membrane and then reach the receptor wherever it is. Okay, so what if this cell isn't supposed to carry out that response? Would it have this receptor for that signal? No. So this is one way our cells can vary, right? They might have different receptors for different hormones or ligands, right? Okay. So the next process here, right, transduction, again involves relay molecules. So they're typically proteins, but not always. We'll look at one example where they're not proteins. But again, they basically take this signal from the receptor, somehow passes the information, and again causes a change here. It causes a shape change. So it's transduced. The signal is transduced. It causes a shape change in the protein, which then causes the next protein to change shape. Then the final protein in that relay will send out for the response. Okay. Okay, so sometimes this is good, right? To have several different molecules, right? Why doesn't just the receptor cause a response right away? Well, if you have several molecules involved, you might have the first molecule might actually go and activate several of the second molecule in the relay, which can then go and activate several more of the third molecule, so you get an amplification of the response. Basically, it'd be kind of like the two of us trying to move every piece of furniture out of here if these were loose, versus me tagging you tagging you, and so forth, so we can all work on something together, right? So you can activate this amplification of different proteins working on a response signaled by the ligand. Okay, can also provide more opportunities for coordination and regulation of cellular responses, and we'll look at how that works, but we can have different inhibitors or activators come in to turn off or on different responses. Okay, so we vaguely hit this, so for anyone who read ahead, can you answer this question of how can a cell quickly activate or deactivate a protein? Phosphorylation. So we haven't really gone that much into it until now, but if you think about proteins, adding a phosphate group to them is a very simple way of either turning it on or off. Okay, so oftentimes these relay molecules in the transduction pathway are just turned on by phosphates or off. Okay, the protein kinases are particular proteins that add these phosphates. Okay, so it's a protein that adds a phosphate to another protein. Protein phosphatases actually remove those proteins, phosphates off of proteins. Okay, so again, a lot of times these transduction pathways are going to be protein kinases. So this is easier understood with a picture otherwise, right? So here we have our transmembrane protein that with the receptor, it got the signal molecule, and then its first response is to turn on this kinase protein here, okay? So once this receives a signal, it turns this kinase on. So this is a protein that will phosphorylate another protein, okay? So once this one is phosphorylated, you can see it has a shape change, right? It goes from oval to circular, okay? So it's signifying that there's a shape change that's active, and it's going to actually phosphorylate the next protein in the transduction pathway, which happens to also be a kinase. So this protein is going to go phosphorylate another protein once it's activated. So again, it goes from oval to circular, signifying that it's been transduced, right? It's caused a shape change in the protein. And this is a phosphorylation cascade because all of these proteins in the transduction pathway are kinases. They're all phosphorylating the next protein down the line to turn it on, to activate it, okay? Okay, so again, it can be amplified if this turns on several of these, right? And then this one would turn on several of those. So you can have amplification that way too. But this is a very quick way to activate this transduction pathway. And then, as it shows here, this last kinase activates this final protein in the pathway that is not a kinase, right? Necessarily. And once it's activated, it will carry out the cellular response, whatever that is. Whether it's rearranging the cytoskeleton, turning on or off a gene, whatever that is, it will send a response. Okay, so how could this pathway be inactivated if these are all kinases? I know I just threw the term at you, but it's through the protein phosphatases by dephosphorylating them, right? By taking those phosphates off, this can be turned off. So here we have protein phosphatases that will go in and deactivate these proteins, right? It turns them back into these inactive protein kinases. And that just wait in the cell, right, until another signal molecule comes in and reactivates the pathway again. So a lot of times, these signals aren't necessarily on or off. It could be a degree, depending on how many kinases or phosphatases are present in the cell. In fact, we have a large portion of our genes that specifically are dedicated to making kinases and phosphatases, because they're very important in carrying out cellular communication. Okay, so don't get bogged down in all this. I'm not even really going to, require you to know what an example of a second messenger, but I want you to know that sometimes in this transduction pathway, there are molecules involved that are not proteins, and they're usually, small ions that can move quickly through the cell. So sometimes you have, you want to activate a process that's deeper in the cell, so that transmembrane protein will send what's called a second messenger to go activate that transduction pathway. So again, just an example, I don't know if it's a G protein receptor or anything, but this might activate the second messenger that can move really quickly and go deep into the cell to activate this protein kinase, which then starts the rest of the transduction pathway to get the response. So again, second messengers are non-protein molecules that are small, typically ions that can move quickly through the cell. Okay, so responses, well, could be a lot of different things, but gene expression, cell growth, division, locomotion, right, metabolism, so turning on or off cellular respiration can be, or activating cellular respiration can be done by this. Secretion, so again, like if you cut yourself, you need to secrete, if your body needs different cells to secrete different things, that could be a response. And then apoptosis, or cell death, are some examples. Okay, these responses though occur either in the nucleus or in the cytoplasm, and then they may be released, right? So if it's to be something to be secreted, it might be released, but the response occurs first either in the nucleus or the cytoplasm. So, for example, like I said, well, this isn't that one, but it might be for gene expression. So when this receives the signal, it sends a transduction pathway, so phosphorylation cascade occurs, and then this protein is able to go inside the nucleus and activate what's called transcription factor. So you guys we haven't gone over genetics yet. I'm not going to ask you as detailed of a question, but for example, just so you can understand how this works, this would be activated, and then it will read your DNA and transcribe that to RNA, which will be turned into a protein. So that's one example. Also, right, so that first picture I showed you was a cheetah chasing a gazelle. So when you need a burst of energy, animals store our energy as glycogen, you remember, right? That starch glycogen in our cells will be activated to be broken down really quickly, so we can have a lot of glucose to break down from cellular respiration to generate ATP, so that gazelle can run really fast, right? So this is actually an example of amplification. I don't expect you to know all this, but the signal will be received, and it goes through. It actually has a second messenger that sends, activates kinases, but again, they activate several other. It amplifies the response, and you end up getting a lot of glucose release. Yes.
So at what point is the amplification?